Hello, everyone. Welcome to today's live broadcast, Significantly Simplify Your Plant Metabolomics Workflow. I am Brenda Kelly-Kim of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by SciEx. SciEx helps to improve the world we live in by enabling scientists and laboratory analysts to find answers to the complex analytical challenges they face. For more information, please visit www.sciex.com. Before we begin, we have a few announcements. This webcast is designed to be interactive, so let's hear from you. You can submit questions or comments by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. Answers always welcome too. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem to the green Q&A button, lower left. If you look at your screen now, you should see two polling questions coming up. The first is, how are you currently analyzing metabolomic samples? And the second is, what improvements would you like to see when analyzing metabolomic samples? If you could take a minute to answer those questions, we would really appreciate it. I would now like to introduce today's speaker, Dr. Stephen Locke. Dr. Stephen Locke obtained his PhD in physical organic chemistry from the University College of Swansea in 1993 and is a chartered chemist and a member of the Royal Society of Chemistry. Previously, a lecturer of chemistry at the University of Hull and a senior scientist at CCFRA, he has worked for SciEx in various technical roles for over 16 years. He has over 25 years experience in analytical chemistry and has presented at over 50 international meetings around the world. Steve is currently Business Development Manager for the Separation Division of SciEx, covering Europe, the Middle East, Africa, and India. I will now turn it over to Dr. Locke for his presentation.
Thanks, Brenda, for the kind introduction, and thank you everyone for attending today. Today's presentation is going to be on the use of CESI-MS in the analysis of metabolomic samples. I'm just going to move to the next slide. So the talk outline, basically what I'll start with is going through what CESI-MS is, and then we're going to look at examples where we can apply this into metabolomics. I'm going to focus on two main areas. The first one is the application of CESI-MS in cationic metabolomics, and the next one is the application of CESI-MS into anionic metabolomics, i.e. negatively charged species. So what is CSIMS? Well, actually it's the connection of capillary electrophoresis to mass spectroscopy via the electrospray interface. So it's basically the seamless connection of CE with mass spec. We do this by physically connecting the capillary coming straight out of the CE instrument to a nanospray source connected to a mass spectrometer. In this diagram here, what you see is the instrument, which is the CE instrument, CSI 8000, and then you can see an arrow which connects it to a triple top instrument. The triple top is an accurate mass QTOF instrument from SciEx. And it stands for the integration of capillary electrophoresis with electrospray ionization into a single dynamic process within the same device. So basically, it really is the simple plug-and-play connection of CE to mass spec without the addition of any other modifiers or gases. It's just a plug-in connection of CE to mass spec. And one of the benefits of doing this is that you can actually then run the CE at its flow rate directly into the mass spectrometer. Now, this has huge benefits in the area of ion generation. For those people who have used mass spectroscopy over the years, everyone knows that there's an increase in sensitivity when you decrease the flow rate of your analyte into the mass spectrometer. And this slide here shows the general decrease in flow rate with the technique. So you can see going from high flow where you use 2.1 to 4.6 millimeter columns. Here the flow rates are in the region of 200 microliters per minute up to a couple of mils per minute. And then you go right down in flow rates down to nano LC, which is in the order of a couple of hundred nanoliters per minute, and then to CSIMS. Here you're running at low nanoliters per minute. So it actually connects the CE directly to the mass spec, and these ultra-low flow rates then go straight into the mass spectrometer via the nanospray source. This happens in the OptiMS capillary, so this is the connection to the mass spec from the CE instrument, and this is where it really happens, the science behind the connection. So in this connection, what we have is the capillary running from the CE, and this is the same capillary you load your sample into and apply the separation voltage. It runs through this OptiMS cartridge, and then it goes through an area which contains conductive liquid. So it's like electrospray ionization, where you have a hollow tube into which another capillary runs. When you're doing a standard electrospray ionization, you have a gas around that tube to generate the spray. Here we apply a liquid. The liquid is conductive liquid, and that transfers the ion spray voltage to the capillary to generate the spray. At these low flow rates, you don't need gas, you don't need makeup liquid. All you need is the application of a voltage to generate the spray for electrospray, and this is exactly how we do it. So, and this means we can run the CE flow without mixing directly into the mass spec. And the capillary length used is standard in these cartridges, so it's about 91 centimeters, and we use a narrow internal diameter 30 micrometer diameter, and this is all patented technology owned by SciEx, and then based on a paper in Analytical Chemistry in 2007. The reason it works so well is that these ultra low flow rates, there's less competition for charge for your compounds, so your effective ion generation is dramatically increased. Also, what you have less to compete with is the loss of solvent, because you have less solvent flowing into the instrument. All this gives you huge benefits in ion generation, and it means that you can, even with the low volumes that you inject onto these narrow capillaries in CE, you still get very good sensitivity, an excellent peak shape of the CE. And this sensitivity is shown in this next slide. This is a bar graph showing the change in ion generation when you decrease the flow rates. So you can see from the right-hand side here, which is at 350 nanoliters per minute, your rough ion generation at infusion is around about a couple e to the 7. However, when you start decreasing the flow rates down towards the left-hand side of this graph, down to where the CESI-MS runs at, you see a massive increase in ion generation, and this really increases the sensitivity of this approach. And it's hugely different to the traditional way of connecting CE to MS, which required sheath flow and sheath gas, which effectively diluted out your sample and decreased the sensitivity of the technique. In this case, there's no dilution of sample. You run the flow directly into the mass spec. And this is all connected via an OptiMS capillary, which fits directly into a Holden and nanospray source. So here we see the implementation of the CSIMS technique when you combine it with a Triplettos instrument. So here you combine the ultra-resolution, high-resolution CE with the high resolution of MS detection. And this instrument, the CE instrument, is also capable, of course, of using standard CE detection techniques as well. So you can, in a way, choose the detection techniques, use your CE and UV detection mode to do the method development, and then change it to the CSI mode, connect the capillary to the mass spec, and then do MS detection. Now, this has already been applied in proteomics and omics research area, And this slide here shows you a screen capture of some results on an omics research paper, and this was done by Professor Herbert Lindner of the University of Innsbruck in Austria. And what he's done here is just compared nano-LC with CSIMS in the detection of peptides and proteins in a HILIC sample, which is taken from a yeast digest. And here you can see the complementary nature of CSI compared to LC. Now, CSIMS, of course, separates the components of a mixture based on its charge and size, whereas LC separates it based on, typically, on interactions with a stationary phase. So they're totally different ways of separating analytes, and here you can see the benefit of this approach when you're comparing the two techniques. In this case, this is just to show you how CSI and LC are complementary in nature. In these sort of Venn diagrams, what you're seeing is the overlap between the two techniques in the middle, and then you're seeing the enhancement or the detection of additional peptides in red by CSIMS, and the detection of other peptides not picked up by CSIMS in blue, which were picked up by nano-LC. But it does show the complementary nature of these two techniques. And what we can see in the future for omics research, metabolomics and proteomics, is probably people doing both techniques to get full characterization of samples. So how does this apply to metabolomics? Well, because CE is a technique which is based on charge and size separation, really where this technique applies to metabolomics is in the area of polar metabolomics. So this is the polar metabolites. And these are actually a difficult area for LC to analyze, because often you have retention issues on standard reverse phase stationary phases, and this could also have matrix interferences as well. One of the benefits, which I haven't shown here, CSIMS, is that when you're running at these really low flow rates, it also helps with reducing matrix suppression, because there's less competition for charge at these lower flow rates, so it also helps with matrix suppression. So this is an additional benefit of this technique, especially in polar metabolomics, which have issues with matrix suppression because they often elute at the front of reverse phase separations. And there's been some research papers already in this area and using CE in combination with mass spec. And there's been some research papers by the Sogo group in Japan, and also by the group in Leiden University Medical Center and Leiden University, where they've already tried to combine capillary electrophoresis with mass spec in the area of cationic metabolomics. So there's some history behind the use of this approach already in metabolomics. But at the end of last year and the beginning of this year, I was posed by a researcher in Europe who is interested in polar metabolomics and whether the new CSIMS can be used in this technique. And what he asked was some probing questions. The first one was, can he analyze underivatized amino acids as well as anionic metabolites using the same capillary? Because ideally what he wanted was a technique where he didn't have to change in the hardware. He could just flip between methods and run cationic metabolites to look for cation metabolites in one method, and then flip and inject again and look for anionic metabolites using the same capillary and ideally the same macroelectrolyte solution. The other question he asked was, can you detect these metabolites at the physiological relevant levels? And this is specifically in the area of plant metabolomics. Can you also separate isobaric metabolites, which were often just geometric isomers by CSIMS? And here he had some specific questions around phosphorylated sugars, because he has encountered issues of separation when he's tried to do this by HILIC chromatography and also by ion chromatography in connection to mass spectrometry. And finally, can you do all this with low sample consumption? And really this is around the ability to run CSIMS initially on your sample and then use the same sample to apply it to another technique, for example, LCMS. And also, in some metabolomic studies, for example, mouse analysis, sample volume is limited. So this is a specific need for that particular application area. So the first question, the first area I looked at was the application of CESI-MS in the area of detecting cationic metabolites or using positive electrospray ionization. And this is the area really of detecting, in this case, underivatized amino acids as well as other metabolites which are positively charged in electrospray. And actually what I did was took a method that we use for tryptic digest mapping and simply applied this to this area of analysis. So in this case, how you set up the CE for the separation is the first you have to condition the capillary. I did this by using water methanol rinses. Then you load your capillary with a background electrolyte solution. In this case, we used 10% acetic acid. You fill a conductive line, so you enable this circuit to transfer the ion spray voltage to the spray. And then you load your sample. I didn't use electrokinetic injection. I simply used pressure fill of the syringe over a certain period of time. And then you apply your separation voltage to separate your metabolites. In this case, what I also did was I made up my samples in 100 millimolar ammonium acetate so I could do some on-column stacking, using a technique called isotachophoresis. And this basically allowed me to inject more material onto the capillary to get a higher loading volume. For the MS method, I sort of used the QTOF instrument in effectively like a triple quadrupole system. What I actually did was set up dedicated product ion scans, Acromass product ion scans for the amino acids I wanted to detect. And I also added, of course, a screening time of flight single stage mass spec scan to pick up other metabolites in samples. In this case, the total scan time was of the order of 1.2 seconds, and this is due to the fast scanning speed of the 5600 Triplettos instrument, which is capable of doing 50 plus, I think it's 50 product ion scans per second. So it can operate similar to the speeds of a triple, quadruple, or Q-trap system. Regards to setting up the source, of course, you don't need gas to generate the spray. You don't need makeup solution. So the only setting you do to generate the spray is applying an ion spray voltage, and then you just position the spray to get the best sensitivity for your analysis. And here you see some of the data. And this was based on the standard provided to me by the customer. And these are the sort of the levels he expected to see in his plant extracts. And here you see a series of amino acids, all underivatized, and these are separated in the order of, I think it's about 20 minutes was the runtime here, for separation of underivatized amino acids by CSIMS. This has also been run on a triple quadruple system, and for this particular application, we have an application that's already available for people who are interested in underivatized amino acids. You can also see GABA at the bottom as well. So the method itself included other positively charged metabolites as well as mainly underivatized amino acids in this case. So that seemed to work quite well, and this is what I expected, to be honest, for positively charged compounds. But I suppose the major challenge for me was actually flipping this now to connect CSIMS to negative electrospray, because if you look at the literature, there are some questions around the possibilities of doing this. And what I tried to do initially was to use the same methodology as I used for the cationic screen, the same capillary, the same background electrolyte solution, to start to run in negative electrospray. Of course, now you're looking at negatively charged ions, so the thing you have to do here in the method is change the polarity, the separation voltage, to drive your target compounds to the mass spectrometer. So the one thing you do change in the method between the cationic and anionic screen is you change the polarity of the separation voltage and you go to reverse polarity rather than normal. And of course, the other thing you need to do is change the polarity of your mass spectrometer and detect a negative electrospray rather than positive electrospray. And this gives you a breakdown of the method I used to assay samples for negative metabolites. And here you can see the separation voltage is very similar to the positive method I used. I used a pressure injection. In this case, it was a 30-second 5 psi injection of a sample, which is equivalent to 10 to 20 nanoliters a sample on column. The same capillary was used, so for both methods, I've actually used a bare fused silica capillary. The same background electrolyte was used, so surprisingly enough, I never changed the background electrolyte solution. So in this case, I used 10% acetic acid as a background electrolyte. Acetic acid is preferred because it's better for negative electrospray for ion generation. And then finally, the other change in the method really is the movement of the polarity of the separation voltage from normal to reverse phase. For the MS, the main thing here is the change in polarity of detection of electrospray. So you're moving from now to negative mode. I had a very similar setup, so I did dedicated product ion scans for a series of negative metabolites and a single stage TOF scan for detected unknown metabolites. Again, I've tried to keep the cycle time low because of the sharp peaks you get in CE separation, because of the high resolution separation. So again, the cycle time, even with 50 product ion scans and a TOF MS scan, was less than 1.5 seconds in the triple TOF. Again, all I have to set was the ion spray voltage in the source to generate spray, and the position of the source was the same for both methods. The first thing I tried to do was separate two disaccharides which were phosphorylated. These are isobaric, which means they have the same molecular weight and the same empirical formula. So it's ideal to separate these because also some of the major fragmentines are the same. What you tend to see in phosphorylated compounds is that one of the major metabolites, so one of the major fragmentines in MS analysis is the phosphate group. So it will be the same for both of these compounds. But what you can see here is that using this acetic acid as a background electrolyte and using reverse phase polarity, we managed to actually separate these two isobaric phosphorylated disaccharides. The one thing I did here was I tried to increase the speed of sample analysis, so I actually increased the pressure on the separation. So one thing you can do to speed up analysis is apply pressure to the separation as well. And I moved from 0.5 psi to 2 psi pressure applied to the capillary during the separation of metabolites. And here you can see it speeded up and speeded up the separation by approximately 2 minutes. So that's one way you can increase the throughput of your analysis. In this case, the sample was simply diluted into water. So I haven't tried to do isotachophoresis here. This is just straightforward pressure injection of a sample before separation. And one of the key things here, of course, when you do this sort of work is to check on linearity. And I did this by repeat analysis of standards to check to make sure it's linear. And the linear range, so the R squared number I got for the separation, and for both of these two phosphorylated disaccharides, which are important in plant metabolomics, is the order of 0.99. So it was a linear response. And I never used any internal standards at all in this approach. And this is probably one of the most important slides I will show today. One of the biggest, I suppose, needs of this particular area is not the disaccharides, but it's the separation of monophosphorylated sugars, which are important in the glucose cycle and also the energy cycle of plants as well as of animals. And here you can see an example where I've separated 3 phosphorylated sugars. And this is extremely difficult to do. You can't do this by reverse phase chromatography, and people struggle to do this by HILIC and other means to get the resolution you see in this diagram. So what you see here is a separation of glucose 1-phosphate from fructose 1-phosphate from glucose 6-phosphate. The top chromatogram, the top pane, is actually what's called a total ion chromatogram. So this shows you all three compounds in the same pane. Below it, I've separated out the three separate sugars, but you can see that they are separated here. It's not total baseline separation, but you can easily identify the different sugars, phosphorylated sugars, using this methodology. And these are all isobaric as well. And the other problem you have with these three compounds is that they have very similar fragmentation patterns. And you'll see for the fructose 1-phosphate, one of the major fragments ions is actually also present in the glucose 6-phosphate. So you really ideally need to separate these out chromatographically, either using CE or other techniques to properly identify these phosphorylated sugars. And the presence and the identification of these was confirmed by the, of course, standard addition of the individual phosphorylated sugars, which were purchased from Sigmund Aldrich. Again, looking at the linearity for these three phosphorylated monosaccharides, they were all, again, as with the disaccharides, they were linear. So you get an R-squared values of 0.99, 0.998, for example, shown in this slide. Again, for all this work, I have not used any internal standards whatsoever. So this is all based on standards without any internal standards. And this data was processed using Multiquant, which is a software which comes with the CESI-MS mass spectrometers. Of course, we need to apply this to real extracts. So actually, the customer supplied me with a wheat extract. The wheat extract, however, was developed for HILIC chromatography, so it actually contains a lot of methanol. In this sort of analysis, you really want to run these in more aqueous extracts. So the prep wasn't designed for CE analysis, designed for HILIC analysis. So I actually diluted the real plant extract by a factor of 8, and then analyzed it and compared it with a standard. And I could actually identify two phosphorylated sugars in the plant extract and separate the glucose 1-phosphate from the fructose 1-phosphate. And here you can see the wheat extract compared with the standard. What you can see also, if you look closely, is the migration times are very, very similar, comparing a standard to wheat extract, and virtually the same. This aids in the discrimination of these two compounds. So that's looking at phosphorylated sugars. Of course, we have the citric acid cycle, which is also important in polar metabolomics. And here is an example separating two of the compounds which are part of the citric acid cycle. And this is citrate and isocitrate. In this example, if you look at the bottom two panes of this picture, you can see citrate and isocitrate, again, are geometrical isomers. In this case, the position of the hydroxy group moves across the molecule, and that causes a different isoelectric point or pKa of the compounds, and you can separate these quite well by CSIMS, and you get baseline resolution of these two important metabolites in the citric acid cycle. Okay, and that's shown in the top pane of the electrophoretic separation of these two compounds. Again, using the same method as I applied to the phosphorylated sugars previously. Again, I then start to look at other compounds. These include ascorbate and pyruvate. Again, these are involved in these sort of energy cycles of plants and in metabolomics. And also I looked at, again, standard curves for these materials, for these compounds. And again, you can see for isocitrate and citrate, it's linear. And also with ascorbate and pyruvate, all the calibration lines I saw were linear for a series of standards which were run for these test metabolites. In this case, I tried to base around what were the physiological levels suggested by the customer. Again, one of the questions that is always asked of me is how reproducible is CE? And here you see 3 separate injections of a standard comparing glucose 1-phosphate, sucrose 6-phosphate, and isocitrate. And what you can see from the 3 separate injections is actually the migration times you see from these 3 different metabolites are virtually identical between injections. So the actual CV of migration time is really low. So you're getting very reproducible electropherograms when I repeated this analysis on the same standard. And actually the calibration lines I've shown you during this presentation are from the multiple injection of the same standard and different standards. So these are not just simple one-point calibration lines, they're for multiple injections of the standards. Again, the other question which is often asked is how reproducible intra and intraday statistics can you get for CSI-MS? And this data was acquired in this area by a collaboration we have with Leiden University. Dr. Rawi Ramachur, who's one of the lead scientists there, actually has a CSI instrument, and he actually replicated this data in his institute. And he ran a series of tests to study the inter and intraday statistics for the repeat analysis of samples, metabolomic samples on the system. And here you can see the CVs he achieved. The interday CVs are all less than 12%. Again, we're not using internal standards. These are simply based on repeat analysis without any internal standard markers. And again, the interday migration time CVs are all about 1%, so they're really low. So this shows you some of the repeatability and reproducibility you can get by using CSIMS. And this is data which has been provided kindly by the group at Leiden University, and it's due to be submitted for publication soon. So I just want to go back to some of the questions which were originally posed by the researcher who wanted to see whether CSIMS can be used in the area of metabolomics, specifically around polar metabolomics. So the first question he asked is, can you analyze basically cationic metabolites, for example, underivatized amino acids, as well as anionic metabolites using the same capillary? And from this data you can see, yes, you can, because basically all I've done here is use the same capillary, the same background electrolyte conditions, and just simply change the voltage in the methods to do exactly this. So I proved that you can do that without having to change HPLC columns or mobile phases, which is something you would have to do if you tried to do this by other approaches, for example, LCMS. The next question, which was posed at the beginning of this presentation, was, can you detect metabolites at physiological relevant levels? And again, yes, if you look at the example of the wheat extract, and this is just one example from a series of tests I did, you can see that, yes, we can detect, for example, monophosphorylated sugars in wheat extracts, even when we've diluted the sample 8-fold, which was provided by a research in Germany. And you can see that the linear calibration lines we're achieving for every compound I've tested are all linear, they're not quadratic. So yes, it is reproducible and it is linear, and yes, we can detect at these physiologically relevant levels. Can you separate isobaric metabolites, which are just geometrical isomers, for example, the phosphorylated sugars? And yes, I've shown you can do that. You can not only separate phosphorylated sugars, but also organic acids like isocitrate can be separated. And in the case of citric, isocitrate, and even the phosphorylated disaccharides baseline resolved, separated by using CSIMS. And finally, can you do this with low sample consumption? Well, if you do the math, the actual sample consumption with both injections, it would be the order of less than 50 nanoliters. So you could run your sample in positive cationic metabolomics and negative cationic metabolomics by CSIMS and use the order of 50 nanoliters of your sample, which will then allow you to move your sample to other techniques and repeat the analysis. there's no other technique which uses such a lower volume of sample and allows you the ability to analyze your sample with several different approaches now. So this is just the start of us developing this area at SciEx. I think this is a research project I started about 8 to 9 months ago, and I'm planning to continue this area. And some of the things we're going to try is to look at optimizing the sample preparation for the CSIMS. Ideally, what we would do was not actually prepare our samples as for HILIC chromatography. We would prepare them for CSIMS. So we wouldn't dilute them into methanol, we keep them in aqueous conditions. And then we're also looking in negative mode to think about modifying the sample conditions to see if we can do some isotachophoresis type injections, where we can load more onto the capillary and then get better sensitivity in the method. Of course, we plan to expand this to other metabolomics areas. This initial study was just around these sort of very polar metabolites which are important in plant metabolomics, but again, these sort of metabolites are important in other areas as well, including mouse assays like mouse cerebral spinal fluid testing. We're thinking about applying this to detecting small chain fatty acids, and also to look at human urine for metabolomics as well. So there are several areas now we can start applying this technique to tackling. Finally, I'd like to acknowledge some of the contributors to this presentation. Again, some of the slides I've shown you today were provided by researchers using CSIMS in the laboratories. For the omics slide, I want to first acknowledge Professor Herbert Lindner, who's done a lot of testing in the omics area, especially around histone chemistry and histones in yeast cells and digester histones. And secondly, and more importantly, I want to acknowledge Rawi as well. He has been my sort of co-author and co-worker in this area, and he's actually repeated all this study in his lab in Leiden to confirm the method is transferable, and he got exactly the same results as me. And you can see that some of the reproducibility and repeatability testing was actually done in Rawi's lab in Leiden. So a big thank you to Rawi for the data he supplied me here. So that ends the presentation for today from myself in the area of metabolomics. And now I'm open for questions. So please, if you have any questions, please send them through to me, and I'm happy to answer them now.
Thank you, Dr. Locke, for that informative presentation. Before we get to our questions, I want to remind our audience how to submit them. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. And we'll sure try to get to as many of your questions as we can. Our first question, in the separation of the phosphorylated sugars, how much of each analyte is injected into the capillary?
Well, I suppose you're looking at the injection of low microgram per mil. So because they're low mass, the actual loading volume is the order of 10 nanoliters. So you're probably loading picomoles of material onto the capillary. So the actual loading amount is very low. Yeah. If you compare it with some of the peptide work I've done, in the peptide work I've actually loaded atomoles of material onto the capillary. So the sensitivity you get here is around the low flows of running CSIMS. So that allows you to load low amounts on column and still detect the material.
Great. Thank you for that answer. We do have another question. Do you have to apply any pressure to maintain your electrical contact in neg-ion MS mode with reverse polarity?
It depends on the capillary. In this case, because you're running with bare fused silicate in reverse polarity, you have to apply some pressure because there's a slight what's called electroosmotic flow, which is running away from the mass spec. In the method I used here, I typically applied a couple of psi pressure. With some of the other capillaries and some of the other modes, no, you don't have to apply any pressure. The electroosmotic flow itself is enough to generate the flow you need to detect your ions.
Great. Thank you. Our next question, can I inject larger volumes of extracts in order to increase the LOD?
I think we haven't tried it yet. For loading larger volumes of extracts, what we would do is we'd try to use a process called isotachophoresis, in which case what you do is you actually dilute your sample into a salty solution which allows stacking. That allows you to inject more and fill more of your capillary with sample to increase the sensitivity. The other approach, which has been used by several groups, is to use electrokinetic injection. In this case, what you do is physically apply a voltage to your sample. That drives all your analytes into the capillary, and then you apply a separation voltage. If you do this approach, you can basically suck your sample dry of analytes, and then really increase your sensitivity of your analysis. However, because you're depleting your sample, you have to therefore, in a future injection, run it from a separate well. You can't go back to the same well and analyze the same sample with electrokinetic analysis. So yes, there are ways to increase your sensitivity and load more on.
Great. Our next question, can this be applied in Triplettos 6600 system?
Yes, it can. I've actually used this method on Q-trap systems, on triple quad systems, on QTOF systems, including the 6600. So yes, you can apply it on several different types of mass spectrometers.
Great. Thank you for that. We have another question. The loading volume of CE is low. How does this affect the sensitivity of this technique compared to LC-MS?
Good question. I think really it's all about a combination of ion generation as well as loading. If you look at high flow, you're running there at a couple hundred microliters per minute. Your ion generation is relatively poor because you have to get rid of all the solvent, but you can inject 10 microliters. With CSIMS, basically you're now running at 30 nanoliters per minute, or less in some instances. Here your ion generation is really high, but you're actually injecting a lot less. In reality, the work some of the testing I've recently done, CSIMS can be equivalent to about a 5 microliter injection by high flow LC-MS. So the low volume injection onto the capillary is countered dramatically by the high efficiency of ion generation you get to these low flows.
Great, thank you. Our next question, how easy is it to connect the CESI to a mass spectrometer?
It's very easy. Basically, all you need to have is a nanospray source and an adapter which allows you to connect in the OptiMS capillary. I actually now use a CSI instrument as a nano infusion device over connecting a syringe, because all you simply do is plug in the OptiMS adapter into the holder onto a nanospray device and apply pressure, and you're running. It's really very simple. I can do it in about 5 or 10 minutes typically.
Great, thank you. Our next question, can you connect CESI to different types of mass spectrometers?
Yes, you can. As I mentioned, I've ran CSIMS on Triplettos systems, including the 5600. You can also connect it to 6600, 4600 from AB-Science, but I've also connected as well to Q-trap systems, and you can also connect it to also triple quadruple systems as well. Basically any system which will fit a nanospray source which can hold the adapter for the cartridge, you can then connect it to mass spec.
I see, great. Our next question, are you planning to expand CESI-MS to different metabolomics studies?
Yes, we are. This is probably the first area we've applied to, because there's a lot of questions around this area, which I had from researchers. But yeah, we're planning to apply this to other areas of polar metabolites. We have a list of some, like a couple of hundred metabolites we're planning to try by CSIMS. So I think this is just the start, to be perfectly honest, with this area.
Great, thank you. We have our next question. How repeatable is the separation of the monophosphorylated sugars?
Again, very good question. I repeated this myself in the lab on 2 or 3 different capillaries, but I wanted to doubly check this. So what I did was actually transfer the method to a co-worker, Rawi, who's based in Leiden, who's worked with me on testing the method, and he actually then set it up on his system, and he repeated the separation. So I've confirmed that you can actually physically transfer the method to another institute in another country on a complete new system, and it's fully reproducible.
Great, thank you. I think that's all the time we have for questions. As at the beginning of the presentation, our audience will now see another poll question appearing on the screen that asks, would you be interested in incorporating CESI-MS technology in your laboratory workflow? And if so, what time frame? If you could please take a moment to answer that question, we'll be sure to follow up with you to answer any other questions you might have about the CESI-MS technology. And before we close, I'd also like to thank Dr. Luck for his time today. Dr. Luck, do you have any final comments for our audience?
No, thank you, Brenda. I think for me, I've been running LC-MS instruments for, I would say, 20 years. And I was really surprised how easy it is to connect the systems together. And I would just get anyone to start looking at the papers because it's really easy technology to use. I think you'll see a big increase in this technology moving forward.
Thank you for that, Dr. Luck. I'd also like to thank, of course, our sponsor, SciEx, for making today's webcast possible. And this webinar will be available for on-demand viewing through March of 2016. You'll be receiving an email from LabRoots alerting you when this webcast is available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. Thank you again for joining us today. See you next time. Goodbye.